Hello and welcome to Nikolai's Genetics Lessons, and today's question is what form of RNA binds to both the codon and amino acid? And here four answers to choose from. Imagine that this is message RNA, and this is going to be 5' end and 3' end here, and somewhere here we have ribosome. Ribosome consists of large part and small part, and here we have three sides. One would be A side, P side, and E side. And of course on the message RNA we can find nucleotides. For example, this can be A, U, G here, A, C, and C, U, C, for example. Then here in each this site we can find tRNA that also we call adapter RNA, that looks like this. It resembles clover leaf. It has 3 stems and 3 loops, and let me draw 3 here. So it also has variable stem that you can find here. Each loop here has its own name. So this is going to be T loop, this is going to be D loop, and this loop we call anti-codon loop. So as you see, this would be codons on the message RNA, and here we would find anticodons. So if we have adenine here, then in the anticodon we would find uracil here. If we have uracil here, then here we would find adenine, and if we have guanine here in the anticodon we would find cytosine. And this is, as you know, start codon. So this loop we call anticodon, and base pairing would be very specific. This third position in the anticodon can be variable. In many cases different bases can be found here, and still tRNA would base pair with message RNA. And here what the prime end this is going to be, if we have prime end here, then if we would stretch this tRNA like this end have to be 3' end, and here the smaller one would be 5' end. So 3' end, 5' end, 3' end, and 5' end. So as you see, 3' end is longer than 5' end. We can find following sequence here: A, C, C. Sometimes also next would be A. So, but A, C, C we can find on all of the 3' endings, and we cannot find this sequence in the gene. So this would be later addition to the 3' end, or what we call post-transcription modification. And to this 3' end, unique to this tRNA, amino acid would be added, and these amino acids would form a peptide chain here. So A site would be site of the entrance, P site would be where polypeptide connection would be formed, and E site would be site of the exit. So here we may find a chain, polypeptide chain that would exit ribosome. And by the way, ribosome by itself consists of more than 50 proteins and about 50 ribosomal RNA. So ribosomal RNA just a part of the ribosome. And here is message RNA, answer A, and as you see, the RNA that binds to both codon, here is the 3 codons, and amino acid we call tRNA, and this is answer B. In order to complete our picture here, this adenine would base pair with uracil, uracil and guanine here, and guanine, adenine and guanine here. And also just a note that message RNA grows from 5' end to 3' end, and when ribosome connects to message RNA, it also moves from 5' end to 3' end. So in anticodon, this is going to be the first position, second and third. And as I said, third position we call wobble position, because base pairing at the third position can be not classical, and different bases can be found here, but still tRNA would base pair with codons on the message RNA. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.